I'm just speaking today on behalf of many co-authors. I'm not a geneticist, and I will try to struggle through with what I'm going to present today. This work was done primarily in response to trying to differentiate whether or not short-jaw cisco is a valid species as part of DFO's species at risk process. So most of what I'm going to be talking about is focusing on shallow water forms of cisco, or the lake herring, and then a deeper benthic form of a short-jaw cisco, although we did certainly do the genetic work on the other ecomorphotypes as we came across them. Just as a short introduction, as I'm sure many of you know, ciscos have only been in these lakes, these large north temperate lakes in North America for about 10,000 years since the retreat of the glaciers, but since then they've managed to display a phenomenal amount of diversity. And this is a famous picture of the 6 species or ecomorphotypes from Lake Michigan. And we also know that in the Great Lakes in particular, we've lost many of these ecomorphotypes. In Lake Michigan and Lake Huron, we only have one benthic form, the bloater now, and some remnant populations of shallow cisco forms. In Lake Superior, we've managed to maintain all of our morphs, but down in Lake Erie and Lake Ontario, they're in even worse shape, where they've basically been reduced to very small remnant populations of the shallow water cisco form. And part of the issue is many of these species are listed or in the process of being listed, but people are uncertain as to whether or not they are actual classic species or whether or not they're just forms, and whether or not we need to protect those different forms based on some of this taxonomic uncertainty. So this work was initiated to try to bring the most recent most recent genetic techniques to bear upon this question about whether these are species and what type of speciation might be occurring in order to bring about some of this fantastic diversity that we see. So what we did in this study was we took fish from 20 different lakes across the range of where we see short-jaw cisco in North America, so from Great Lakes up through the large lakes in the Northwest Territories of Canada. We analyzed their genetics at 290 AFLP loci and did this work on quite a large number of individuals, almost 1,400 different fish. And we tried to assess 3 different hypotheses for the diversity and origin of these fish: the plasticity hypothesis, the good species hypothesis, and something called the parallel origin hypothesis. And I'm going to briefly introduce those now. So the plasticity hypothesis is the simplest hypothesis. It's one that just suggests there's only one lineage of ciscos, and that within each lake where we see different ecomorphotypes, the diversity we see is a result of the fish simply living in different environments and taking on an ecomorphotype that's advantageous for the area that it's living in the lake. So for example, fish are born, and if some of them end up in the deeper part of the lake, they take on the characteristics that will be most successful for them in that part of the lake. So few gill rakers able to pursue a benthic lifestyle. The good species hypothesis suggests that fish were differentiated prior to glaciation, so there was a shallow water form and a deep water form before the glaciers retreated, and that these fish then moved out into all the lakes and repopulated them as true species or good species at that time. And then finally, we have the parallel origin hypothesis, which assumes that these ecomorphotypes originated only recently and independently in each of the lakes. So there was a single genetic type that went out, was distributed by the as the glaciers retreated, and we had these shallow and deep water forms repeat themselves independently in each of the lakes where we now find them. And so each of these different hypotheses come with contrasting predictions. The plasticity hypothesis suggests that genetic clusters should only correspond to lakes. There will likely only be a single overall single lineage that we can identify based on the genetics, and that the last one here is the most important one, that ecomorphotypes will not form distinct gene pools within lakes, so there will be no genetic differentiation. The good species hypothesis is probably the most different one. It suggests there'll be several independent lineages, and that genetic clusters will then correspond to these ecomorphotypes, and that regardless of where you capture the fish, samples of a given ecomorphotype will more represent, their genetics will more represent the similar ecomorphotypes from other lakes than within other ecomorphotypes within the same lake. And finally, the parallel origin hypothesis is similar to the first, the plasticity hypothesis, in our predictions, with the exception of the last one, where samples of different ecomorphotypes from the same lake should then be more similar to those of other ecomorphotypes within that lake than from between lakes. And so we have, I think, now a really good data set to contrast those predictions. As I mentioned, we use AFLP markers. We had data from 20 lakes, including some of the lakes. We had several areas within lakes, and we had pretty good sample sizes. 10 of our lakes had short-jaw cisco and the shallow water cisco form in them, and we had fish from pretty much across the range, from Lake Ontario through Lake Huron and Lake Michigan. Good data sets from Lake Superior, Lake Nipigon, and then some of the smaller inland lakes as well, and then up through Great Bear, Great Slave Lake, and some Lake Anna. Well, I'm not going to try to pronounce it, in northern Manitoba. So we had good spatial coverage and good sample sizes. And Julie and her grad student working on this used some common genetic techniques, the program's structure and flock, to look for patterns in the genetic data. And then they used a number of other analyses similar to discriminant function analyses and analyses of molecular variants to address some of the questions. So I'm going to jump right into the results. This is looking at all of the lakes and all of the fish, and each, I guess, should explain these figures a bit. Each fish gets a bar, and these dark lines separate the lakes. So each individual fish, the ecomorphotype or the genetic pattern is on the bottom. So here we have, for example, on the left-hand side, fish from Great Bear Lake. And we have different gillraker forms. So this is White Partridge Lake in Algonquin Park. We have a low gillraker form. In the middle here we have the fish from Lake Nipigon and the Great Lakes, different localities. And each fish gets a bar, and these are the genotypes that have come out of the AFLPs that we used for this analysis. So what we see, and the way the genetics data comes out, is it tells you the number of clusters based on, or the most parsimonious solution for the number of clusters based on the genotyping that occurs. So when we look at all of the data from across the entire range, we see that there are two clear patterns, or two clear genotypes. We have one that's primarily from the west, other than this single lake in Ontario, and another that encompasses all the lakes from the east. And Lake Nipigon is actually the divide. So all the Lake Nipigon fish show up in this western cluster, and the rest of the Great Lakes in the eastern cluster. Now this actually supports earlier genetic work on the shallow water form of cisco that shows that there were two glacial refuges for these fish. There was a Mississippi and an Atlantic refuge, and the eastern fish are primarily dominated by this Atlantic refuge, and the western fish by this Mississippi refuge. So that's what this first series of results shows. The second way that the data can be parsed, again, this is the whole data set, comes up with 15 clusters. 12 of these clusters are related to individual lakes, and I'm going to touch on some of what I think the key ones are here. You have some of the smaller inland lakes are clearly different. You have this large purple blob in the middle here. These are fish from all of the upper Great Lakes. Lake Ontario is quite interesting, and I'll talk about that in a little bit as well, but it actually comes out as its own unique genetic structure from there. And out of all of these, there's only a single cluster that's related to its own ecomorphotype. So the fish from Lake Nipigon are here, these pinky sort of fish, and in both we fished two different areas of that lake, or analyzed fish from two different areas, and this lightest pink color are actually the short-jaw cisco ecomorph from Lake Nipigon, and those are the only ones that show up as being different in terms of ecomorphotypes for this broad analysis. The rest of these colors and clusters relate to individual lakes, not ecomorphotypes. When we look specifically at the Great Lakes, again, there are the best solution is just two distinct clusters, and as you would have seen from the very first graph I put up, that's because we have strong differences between Lake Nipigon and then the rest of the Great Lakes, including Lake Ontario, which is more closely aligned to the Great Lakes. Now, the second way to look at the Great Lakes data, so if you ignore sort of that first level clustering based on the glacial refuges, it gets a little more interesting. You see a number of clusters, both based on the lakes and from the ecomorphotypes themselves. So the first thing is these green fish here are Lake Ontario, again, clearly different from everything else that we see in the Great Lakes. Second thing to note is that in Lake Nipigon, again, you have these clusters that are based on ecomorphotypes. So we have a short jaw cluster from the north and south areas, and we also have blackfin cisco, which is a species we don't talk about much in the Great Lakes anymore, but both the blackfin clusters separate out here as being unique and distinct. And then the remaining Lake Nipigon fish, again, are more similar to each other than everything else. Unfortunately, when you get to the upper Great Lakes, so Lake Huron over here and Lake Michigan, and then Lake Superior, we have data from many different areas. The patterns kind of fall apart and everything looks very much the same. They are similar to each other, but they are, in terms of ecomorphotypes, there doesn't seem to be much of a pattern. And this kind of bears out when you start doing some of the genetic reallocations. So this is, again, similar to a discriminant function analysis reallocation, and you can see quite clearly that Lake Nipigon fish are almost always reassigned back to their lake of origin. Same with the Lake Ontario fish, but for the other fish it's much less. Lake Superior fish and Lake Huron fish show up here as being quite similar. Lake Nipigon fish, or Lake Michigan fish, we don't have many fish, but they're pretty much randomly almost distributed, but as you would expect, much more similarity between Huron and Michigan. And even in Lake Superior, we have quite a few fish being assigned to either Lake Nipigon, or sorry, Lake Michigan or Lake Nipigon. When we look at morphotypes within the Great Lakes, it's even worse, other than some evidence for blackfin and kai to being unique ecomorphotypes, but the remaining three species, bloater, cisco, and shortjaw cisco, are just almost as likely to be assigned to any of those other morphs as they are to what we originally would have classified them as. Honing in specifically on Lake Superior now, the best solution was actually only a single genetic stock, or a single genetic composition. If we force it to two and you really squint, you can kind of see some patterns with kai and short jaw having more of these pinky bars than the blue bars, particularly in comparison to the Lake Cisco or Cisco forms. And part of the issue might be that there seem to be differences between the eastern and western part of the lake. So we split up our Lake Superior data set with fish caught from the east and the west, and you can see that in the eastern part of the lake there really is no discrimination among the different forms. We see very low classification rates back to what we would have identified them in the field. In the western part of the lake, things are a little different and a little better, particularly for the short jaw form and the kai form, where we have a little bit better chance of correctly reassigning those genetically along back to the ecomorphotype at which we identified them. So when you look at the Great Lakes picture, it looks like pretty much there is genetic homogeneity. But when you look outside of the Great Lakes, and in addition to Lake Nipigon, there are quite a few lakes where we do have very distinct differences between the shallow water cisco form and the deeper water short jaw forms. So for Trout Lake, White Partridge Lake, Lake Athapasco, and Great Bear Lake are 4 of the lakes in addition to Lake Nipigon where we have these distinct differences between these ecomorphotypes. So there are possibilities to pick up these differences when in fact they exist. So half of the lakes that we had short jaw in, we were able to get clear differentiation. So to summarize, our data shows that we have 2 broad genetic groups, but that these groups are not based on ecomorphotypes as we would have expected given some of our different hypotheses, but instead they're based on different glacial refugia. There is evidence for differentiation, genetic differentiation among the Great Lakes. Lake Nipigon is clearly different than the other lakes, and so is Lake Ontario. But Lake Superior, Huron, and Michigan have very similar genetic composition to the Cisco communities. Within lakes, short jaw and blackfin Cisco are clearly differentiated in Lake Nipigon, and in Lake Superior, short jaw Cisco and Kaiai in the western basin appear to have some evidence for genetic differentiation, but in the eastern part of Lake Superior, again, there is pretty high genetic homogeneity. And no other ecomorphotypes were supported in Lake Huron, Lake Michigan, or, as I mentioned, the eastern part of Lake Superior. So if we go back to my original predictions to see which of the hypotheses are most supported, for all of the different hypotheses, we do have some issues, although the one that clearly stands out as being the best fit is the parallel origin hypothesis. The plasticity hypothesis, the first 2 predictions are actually generally met, but the third one here in those 5 lakes where we have clear differentiation of the shallow water and deep water forms, we can kind of strike out the plasticity hypothesis. The good species hypothesis, or what historically might have been allopatric speciation hypothesis, all of those predictions were not met in our data set. And that just leaves this last one, the parallel origin hypothesis. And even though this first bullet wasn't always met in all cases, for the most part it was, and these last ones were generally met, at least in most or some of our lakes. So we're left with this parallel origin hypothesis as being the one that is most supported given the data we have. Now, how do we fit this into a recovery or restoration framework for the other lakes? I think some of our data is important. We know that there are efforts to restore deepwater Ciscos to Lake Ontario that are ongoing. And if you remember back, it's clear that Lake Ontario is quite different genetically than any of the upper lakes. I think that's something that managers should place in the back of their minds as we move forward with that restoration. The second thing is that it's obvious that there has been some probably waxing and waning of these gene pools over time. Right now, it looks like there's been quite a bit of introgression in most of the Great Lakes anyway, and so it's possible that to continue to have these forms exist even if you don't have complete genetic isolation, and I think that's an important message to take away as well. So with that, I will try to answer any questions. I tried to use the Henry Quinlan method and